Good afternoon and welcome back here at ECCMID TV, brought live to you from Pavilion 1 here at the ECCMID Congress Center in Lisbon, Portugal. My name is Judith Cohen. I will be hosting these ECCMID TV broadcast series, and I am very delighted to introduce you to my final guest for today. Don't worry, we'll be back tomorrow. And my next guest is Dr. Ursula Theuretzbacher, who is the owner of an independent center for anti-infective agents in Vienna, Austria, but does many, many other things as well, such as coordinating international scientific projects. She manages study groups, publications. She advises regulatory and government agencies, as well as research institutions on antibacterial and antifungal drug treatment. And better known here at ECCMID this year, today she was the chair of the Therapeutics Pipeline Corner, of which I would love to pick her brain a little bit more. So a warm welcome to you, Ursula.
Thank you.
Thank you for coming in here in our live TV studio. So today was the Therapeutics Pipeline Corner. Are you a satisfied chair?
Yes, I'm very happy with the outcome of this session. It's quite popular now. We started this type of session 8 years ago, and the reason for it was to provide academic institutions and small companies with the opportunity to show their contributions to the global pipelines,
yes,
antibacterial pipelines. We depend on them, otherwise we wouldn't get any new supply of new compounds into the clinical pipelines. So without small companies and universities, we sometimes forget about that, we wouldn't get new antibiotics. And most new discovery projects are coming from universities, and most of them are then in-licensed to small companies or spin-offs of universities. So we really need them.
And can you share with us a little bit of the new innovative compounds or developments that you have heard of today in the Pipeline Corner here at ECCMID?
Yeah, so I can tell you we still have innovation. We don't lack innovation.
That's good.
The problem is translation to a drug discovery project.
Yeah.
So my opinion is that we need certain requirements for a good drug discovery project.
What kind of requirements are you thinking of?
So very important nowadays, because we have the resistance problem, and we need to address resistance. We have a lot of old antibiotics that work perfectly, but not if we have resistance. So we need new drugs against resistance, our resistance problem. That means we need novelty. We need completely new approaches. So that could be new chemical or functional classes, new targets, new mode of action. The goal is always to avoid cross or co-resistance with old antibiotics, because we need something in case...
The pathogen should not be able to recognize a new compound at all.
Exactly. So that's the goal. That's why we need novelty. Not because just we like innovation, but the innovation to avoid this cross-resistance and to have something against the most resistant bacteria. But innovation is not enough. So it also needs to have clinical utility. There's a lot of innovation, but without really clinical utility. I mean, it doesn't benefit the patient or patient population, so it needs to have clinical benefit.
And why is that? Is that because in later development phases they fail to reach the effectivity or the effectiveness that was
or it's
anticipated?
It's just useful in a very specific, rare situation, or in a very, very small patient group.
population. Yeah.
So it needs, you have to be able to show clinical utility.
Relevance, yeah.
And that leads us to the next topic, this social value. Here we have the tension between the individual patient, so the very popular concept of individualized, personalized treatment that we know all from cancer. For infectious diseases, it's different because here it's about usually acute infections. You cannot wait for long to have all diagnostics that you need in place.
Unless the diagnostics innovations up their game and get us rapid testing.
Yes, but it's
I
also
understand
here
it.
it's not only about
Yeah.
innovation.
Yeah.
Also this field is
Yeah.
innovative, but it has to be used in clinical practice. And if it requires a really sophisticated diagnostic infrastructure that's only available in very specific places, then we need to
It's not of
discuss.
great use.
We need to discuss what society really wants. Do we want new drugs for very few patients, or do we want drugs for a patient population that's also usable for empiric therapy?
I get it.
We still need empiric therapy for situations where we expect high resistance and where we need to treat immediately. So not being able to wait a day or two even. So that's a little bit of a tension between the individualized patient aspect and public health aspect. So I'm coming always from the public health perspective and not so much from the individual patient.
And was that reflected in the selection of contributors to this year's ECCMID Therapeutic Pipeline Corner?
Yes, I will come to this aspect soon. I just wanted to have more
I'm
of
putting the pressure up.
these characteristics that I need is necessary. Of course, you want new antibiotics that don't generate resistance immediately. So emergence of resistance, even during therapy already, should be avoided. And there are concepts where you can avoid this from the very beginning of drug discovery. So it's an aspect that should be considered. And the last, I mean, there are many aspects, but the most important is you have to be able to develop this drug. If you don't have enough patients for clinical trials, or not enough patients afterwards to treat, then it may not be possible to even develop it and get to approval. So these are aspects that really need to be considered very early on, and I see that often as a problem. So there is so much excitement about innovation.
The innovation,
And really very interesting and exciting scientific concepts.
But they're lacking in the clinical relevance and the broader perspective.
Yes, and thinking you almost have to start with a drug discovery project with the patient, so with the end point.
Start with the end in mind.
Yes, and then go back to your drug discovery. But coming back to today's pipeline, since 8 years, I try to always ask new companies, so not repeat myself. And this year, we had quite early approaches and a big variety of approaches. So one company was just brand new, started 2 or 3 months ago.
Oh, that young.
So that was really good and very innovative concepts based on artificial intelligence and and very...
That's very exciting for such a young company to have the opportunity to, at
Yes.
a platform like ECCMID,
Yes.
to network
And that's
with,
exactly
yeah.
why you have the Pipeline Corner, to present them
Yeah.
in this...
to the world.
Yes, in this early stage. Then we had immunotherapies, we had antivirulence approaches. So these are more therapies that are helping patients to get healthier sooner, and they're usually given together with an antibiotic. So you could call them more adjunctive therapies, because we have still infections that we can treat perfectly with good antibiotics, and still we have a high mortality.
Yeah.
So these are approaches where we all hope that the outcome may be improved, despite having good antibiotics.
I understand.
Then we had biologics. So that's, I hope I didn't forget.
Yeah, so the
much.
selection you made for this year's Pipeline Corner here at ECCMID, does it reflect, because you just mentioned that you rather choose for newer companies, so as not to repeat yourself every year. Does the Pipeline Corner, as it was presented today, does it reflect the more global pipeline for therapeutics?
I don't think so, because these were just 7 companies. And one of them, that was a little bit of an exception, was not even a company, but a new incubator or accelerator that supports these young companies with advice, knowledge, but also a little bit of funding. So that was a new approach in Europe, and I thought it would be helpful for small companies to get to know about it, also because it's quite new.
Yeah.
So altogether we just had 6 companies, plus this incubator. And that's not really representing the pipelines, but I think it's still a good snapshot of some approaches. We had small molecules, direct acting, so that are killing or inhibiting bacteria. We had more indirect acting, like the antivirulence. We had large molecules. So it was quite diverse. Yes.
So which global innovation or development, I think I'd rather better call it development, in the therapeutics guideline are you specifically looking forward towards, or do you have good hopes for?
That's very difficult to say in an early stage.
Yeah, I understand.
So usually it's a little bit paradox, but little you know, the more hopes you have that it will be a
progress
really good
to be something
candidate.
of very large
the
value.
more you know, the more you know the challenges. So if you are asking about the global early pipelines, so the pipelines before it gets into the clinical development, this is of course very difficult to see, because they are usually not even in the public domain. I think it was about 3 or 4 years ago, I did a very exciting project that was funded by the German government about the global preclinical pipelines. So I analyzed them, and I had a wonderful source of information that's usually not accessible.
No,
so
I can
these were CARB-X and repair, the impact fund repair. These were applications for funding. So it was confidential information, and of course I did not reveal any single company information, but aggregated and anonymized the data. And these were more than 400 proposals, projects.
Wow, more than 400.
So that gives a little bit of an idea.
Yeah. You're really on the cutting edge of the
Yes.
new developments.
Yes,
Yeah.
so that was really exciting. And I would say it is innovative. We have really good innovation. But if it's possible to translate this innovation into a drug discovery project, a viable drug discovery project, is a big question. So I would say a big part of these projects may not really get anywhere. But there's enthusiasm about the field, there's innovation.
But isn't it so that it's necessary to go through these steps, even though they may fail later on in the process? You need to walk these steps before you know what to discard and what you need to progress with, right?
Absolutely.
Yeah.
And
that also stresses the importance of these kinds of platforms, such as the Pipeline Corner.
Yes. And I even think that we need much more of them, because we know from experience that most of them will fail. That's normal in all fields of drug discovery. And especially now, because we are turning from the, to widen the spectrum of possible approaches. From the beginning of antibiotics to the 2000s or something, so the first 60, 70 years of antibiotic therapy, we had traditional approaches. That means small molecules that were killing or inhibiting certain bacteria. Was very successful. Most of them were natural products that were then modified to address certain problems. Very successful approach. But this approach now has I would not say it came to an end, not at all, but we have challenges. And I think that some companies or let's say researchers think that it would be easier to do something else.
Something completely different.
Yes. I'm not convinced at all that that's easier. But so we have a much bigger variety now. And these are summarized as non-traditional approaches, and it could be phages, it could be antibodies, it could be microbiome modifying, could be immunomodulators, it could be all kinds of antimicrobial peptides. So it's really a much bigger variety now than we had before. That
but it's also gotten more complex.
It's very complex, and it also means we are lacking a lot of basic science.
I see where you're getting.
So it's, it's much less risk to take known antibiotic classes, so have the chemical, right, and then modify it. That means starting at a much later point in drug discovery, and we know a lot of things already. So less risk, but also less reward. Because we are addressing with this approach certain class-specific resistance mechanisms, but never all of them that are important. That's why we need something new. It's challenging, it's really difficult, and we have not solved fundamental scientific questions. And one of the main questions that is still not really solved is how to get a compound into the gram-negative bacterial cell. So we have an envelope that's really tight. And bacteria don't like substances to get in.
No.
So they are either protecting themselves or pump them out again.
Yeah.
And this is really difficult to overcome.
Yeah.
So still a lot of science to do.
A lot of science to do. Back to the basics, but also back into or innovating.
Yes,
Yeah, so
yes,
it's the combination that is the strongest.
Yes, and we need that
Yeah.
because otherwise we don't get clinical pipeline drugs.
Yeah.
We see that the current clinical pipelines are really not encouraging.
Yeah.
We get, and I'm sure that they will come, they will get to the patient, but these are all modified molecules of old classes. So what I just said, they are addressing a very specific resistance mechanism, but not solving our problem of the most resistant bacteria. And this clinical, it will take years until we get something new that has no cross resistance. So the next 10 years,
You're not expecting
I'm not expecting
much progressing to that phase, at least. A lot. You'll probably be expecting a lot new.
yes,
or a lot of
we
innovative
don't know
concepts.
if it will get
Progress.
to this
I
stage.
understand. Yeah.
I think we still, the basis of drug discovery, so the number of companies, projects that are existing is too small.
Yeah.
This is a problem of the entire field. So we don't have enough funding.
Yeah.
There's not enough money.
Yeah.
Expertise has been lost.
Yeah.
So when the big
But the risk is quite great that
the risk is
the
not
expensive things that you are developing will
yeah,
not
but
lead
you have
to
always a risk when you are developing whatever drug.
Yeah.
Drug discovery is a risky business.
Absolutely.
And that's the same for antibiotics.
Yeah.
When you are at a certain stage, then the risk gets, of course, lower because you have PK/PD studies where you can predict the dose, and you can predict a lot of things with modeling. But in the very beginning, it's hard.
Yeah.
It's really hard.
Yeah. To progress from the unknown to the known is a difficult one.
Yes.
Well, I think we're running out of time, but thank you so much for giving this extra insight on, the complexity of innovation and the creation of new therapeutics in the field of antimicrobials. I think it's very interesting, and you've really stressed the need for an ECCMID program element such as the Therapeutics Pipeline Corner. So thank you very much, Ursula, for coming here and taking the time to elucidating this some more. Thank you all for watching and tuning in with us again here at ECCMID TV. This was our final broadcast for today, but we will be back tomorrow morning. We will start 1 hour earlier than you can expect from the past couple of days that we did. We will be starting at 8 o'clock when Jacob Morangelad, ECCMID's program director, will be presenting to you his personal selection of highlights for tomorrow, which is already the final day of ECCMID. So please tune in with us tomorrow at 8 o'clock. We'll be there fair and square. See you then.